Thin layer chromatography and column chromatography are the two most common chromatographic methods for the separation of mixtures of organic compounds. TLC is an analytical method, whereby a small portion of a sample is separated into its components for sample analysis only, not for separation of the entire sample. Column chromatography is a preparative method, used to separate most or all of a sample into its components for further use, as a reactant in a chemical synthesis, for example, or for further analysis of the individual components. The method involves partially filling a glass or plastic column with the stationary phase, loading the sample at the top of the column, then pouring the mobile phase into the top of the column. As the mobile phase slowly percolates down the column, chromatographic separation of the individual components in the sample is achieved by exploiting the differences in affinity, or stickiness, between the mixture components and the polar stationary phase. Those components least attracted to the stationary phase will spend more time in the mobile phase, and therefore will move down the column fastest, while those components most attracted to the stationary phase will spend less time in the mobile phase and will move down the column at a slower rate. TLC and column chromatography are chromatographic methods, so each employ a stationary phase and a mobile phase. In TLC, the mobile phase moves up the stationary phase by capillary action, and in column chromatography, the mobile phase moves down the stationary phase by gravity. In TLC, only one eluent is used, but in column chromatography, while you're running the column, you can gradually change to eluents of increasing eleotropic strength in order to maximize separation of components. An example of chromatographic separation by column chromatography is illustrated in this animation, where a mixture of three components, one very polar, one of medium polarity, and one nonpolar, are adsorbed to the top of a silica gel column, which was prepared by filling the column with silica gel and then completely wetting it with pure hydrocarbon solvent to pack it tightly. The first eluent added to the column is a mixture of hydrocarbon, which is very nonpolar, and ethyl acetate, which is much more polar. More polar solvents are said to have a higher eleotropic strength, or more simply, a higher ability to elute or move components along a stationary phase. Since this mixture is 90% hydrocarbon, its eleotropic strength is only slightly greater than pure hydrocarbon, so the only component mixture to move far is the nonpolar component, since it doesn't stick very strongly to the very polar surface of the silica gel. Eventually, by adding more of that eluent, the nonpolar component elutes off the column and is collected in column fraction number 2. Fraction number 1 is what's referred to as a dead fraction, since it contains only the mobile phase that came off the column with nothing dissolved in it. The component of medium polarity continues to elute down the column, but much more slowly since it's more strongly attracted to the stationary phase. Eventually, that too elutes off the column and is collected in a separate test tube labeled fraction number 4. The polar component, being strongly adsorbed to the polar surface of the silica gel stationary phase, has moved very little up to this point, and it takes a much more polar solvent with a higher eleotropic strength to get it to move down and eventually off the column, in this case pure ethyl acetate. In the experiment demonstrated in this video, the pigments isolated from a leafy green vegetable will be separated by column chromatography based on their differences in adsorptivity, or their ability to stick, to the polar stationary phase. The two broad classes of pigments isolated can be identified by their color and the order in which they elute from the column. The pigments are isolated from the dried plant leaves by solid liquid extraction. This is achieved by grinding the dried leaves in the presence of a suitable organic solvent. The pigments extracted fall into two broad categories: carotenoids, which are yellow to orange in color, and chlorophylls, which are green to yellow-green in color. Also present are pheophytins, which are basically chlorophylls without the magnesium ion, and they are blue-green to gray in color. Begin with the pigment extraction. 50 milligrams of dried parsley leaves are used in this demo. If the dried leaf flakes are large, as in this case, briefly grind them into smaller fragments, as shown here, to increase the solid surface area for a more efficient extraction. To the tube containing the dried leaves, add approximately 1/2 milliliter of a hydrocarbon solvent such as Lagroin, in this case 100% heptane. Then add 3 drops of acetone to the tube. The solid liquid extraction is performed by using one hand to securely hold the bottom of the tube while keeping the tube bottom resting firmly on the benchtop surface. In the other hand, use a sturdy glass stirring rod to grind and crush the suspension of plant material and solvent. The rod should be held close to the tube opening while grinding, and excessive force should be avoided to prevent puncturing the tube or snapping the glass rod. Do this for a full 2 minutes. This should result in a dark green suspension. Cork it and momentarily set it aside standing upright. A short pipette microcolumn is used for this relatively small-scale separation. Insert a very small plug of cotton into a short-stemmed pasture pipette. Use a boiling stick as a probe to lodge the cotton where the pipette narrows. Do not pack it too tightly, otherwise the flow of liquid from the column will be very slow. Above the cotton, add a very small layer of sand. Above the sand, add approximately 0.75 grams of silica gel. Gently tap the pipette briefly on a soft surface to allow the silica to pack tightly. As you do this, you can see the height of the silica gel in the pipette decreasing. Then, cover the silica gel with a very small layer of sand. Use a 3-pronged clamp and clamp holder attached to a ring stand to support the column. Fill the empty space above the silica with Legroin. Use either a rubber bulb or this syringe pressurization device to pack the column by forcing Legroin through the silica gel, which packs the stationary phase tightly to achieve better separation of pigments. Force the liquid through the silica gel into a 25 milliliter Erlenmeyer flask until the entire column is wet and the liquid level just reaches the sand above the silica gel. Add more Legroin as needed. Do not push the glass of the pipette column too far into the tube of the syringe apparatus or rubber bulb if you're using that. You need to be able to quickly remove it to release pressure just as the descending liquid approaches the sand. Don't forget to remove the rubber bulb or air pressurization device from the column before refilling it with air. Otherwise, you'll suck the contents of the column up toward the syringe, thereby ruining your column. Repeat this process until all of the silica gel in the column is completely wet. You might have to do this 5 or 6 times in order to completely wet the column and have it packed tightly. Remember that a rubber bulb works just as well as the syringe device to force Legroin through the column. But remember not to push the glass of the pipette column too far into the rubber bulb so that you can quickly remove it to release the pressure just as the descending liquid approaches the sand. When packing the column with Legroin, you can reuse the Legroin that has already gone through the column. Remember to have your other hand ready to help quickly dislodge the rubber bulb just as the descending liquid approaches the sand. Once the column is packed, be sure there is always enough solvent in the column to cover the packing, and as you proceed, never let the column go dry. Add more solvent as needed. The column is now ready to be loaded with the pigment extract. Use a pipette and rubber bulb to transfer the green liquid from the test tube onto the column. Squeeze air from the bulb. Place the tip of the pipette flat against the inner bottom of the tube, then carefully draw up the liquid away from the undissolved plant material and add it to the top of the column. Let the extract go just below the level of the sand. You can carefully force it down using air pressure from a rubber bulb. Remember to release the pressure just as the liquid approaches the sand. At this point, you don't want any portion of the column to become dry. Then, wash down any pigments on the inner surface of the column with small amounts of Lagroin. Repeat this several times more, each time letting the solvent go just below the level of the sand before adding the next portion of Lagroin. In this way, you ensure all pigments will be stuck to the surface of the silica before you fill the column with eluent. Even at this point, you can begin to see some yellow pigment separating away down the column from green pigment. Now that the pigment is completely loaded on the column, you can fill the empty space above the silica with a solution that is 9 parts Lagroin and 1 part ethyl acetate, the first eluent. Let the column slowly drain by gravity into a clean, dry 25 milliliter Erlenmeyer flask. As you watch the two major groupings of colored bands of pigment elute from the column, attempt to collect all yellow bands of color into one small test tube and all green bands of color into another small test tube, while collecting the dead volumes, that is the clear liquid where no colored compounds are eluting, into the 25 ml Erlenmeyer flask. Try not to collect any of the dead volumes into the tubes containing the colored pigments, because that will dilute the colored fractions, making them less intense in color. Depending on how tightly the column was packed, how much cotton you used to make the column, and how tightly you pushed it into the pipette, the first color band of pigment could take 20 minutes or more to elute from the column. If the yellow band has eluted down the column by gravity in a relatively tight band, as in this example, you can use the pressure from a rubber bulb to make it elute more rapidly once the band has reached approximately 1/2 to 2/3 of its way down the column. When the band has reached the sand at the bottom of the column, use the pressure from a rubber bulb to hasten its exit from the column into a small test tube. Continue to apply pressure with the rubber bulb until all of the yellow pigment has eluted from the column. You can see that no more of the yellow pigment remains on the column. Now fill the empty space above the silica with 100% ethyl acetate, the second eluent, to elute the more polar pigments that better adhere to the stationary phase. After adding ethyl acetate, carefully use air pressure from either a rubber bulb or the syringe Tygon tube apparatus to force all the remaining pigments down and off the column. Add more ethyl acetate as needed and don't let the column go dry. Using air pressure, the second pigment should elute very rapidly. Collect it in a separate test tube. Here you can clearly see the results of column chromatographic separation of the carotenoid and chlorophyll pigments extracted from parsley. Instead of using either gravity or air pressure to separate and elute pigments from the column, you might be asked to separate the pigments using a technique called dry column vacuum chromatography, which uses vacuum pressure to rapidly pull the eluent down the column, affording a quick and easy separation of a mixture. This technique is demonstrated in a separate video, which you should view regardless of whether you are assigned this method to separate plant pigments.